Mycobacterium tuberculosis causing tuberculosis is one of the most studied bacterial pathogens. Every year, more than 1 million people die and new cases are recorded. And these bacteria have several unique characteristics, such as high GC content of genome, lack of the plasmids, no evidence between strain recombination, and it's really genetically monomorphic microorganism. At the same time, the situation of this tuberculosis remains extremely tense due to the frequent detection of multi-drug and also extensively drug-resistant strains, which leads to an expensive treatment and temporary disability. On this slide, you can see countries, so this high tuberculosis burden, and as you can see, Russia is one of them. And of course, the problem of development of new TB drugs is acute for our country and I think worldwide. But only one new anti-tuberculosis drug, bedaquiline, has been introduced recently. And the process of developing new drugs is a really very, very slow process. In this regard, testing of available medications to search for among them active antimicrobacterial drugs is really important. And as I talked earlier, GC enrichment of mycobacterial genomes also results in the appearance of G quadruplex, also known as G4, which is a spiral G-rich non-canonical form of DNA organization. Scheme you can see on the slide. Indeed, G4s were found in the promoter regions of Mycobacterium tuberculosis, and consequently G4 ligands, as example, BRCA19, inhibit growth of mycobacterial cells. On slide, you can see example of such work and publications. Authors use BRCA19 and really show that it inhibits growth of mycobacterium and stabilized G quadruplex in genome of Mycobacterium tuberculosis. In this fact, we assume that antibiotics of the Aerobic acid group may also be potential G4 ligands. They were now studied as anti-tumor drugs, and they bind to the GC-rich sites of the DNA minor growth. On slide, you can see scheme of these drugs which we use in our study. It's chromomycin, mitramycin, and olivomycin. And in our work, we use the fast-growing and non-pathogenic Mycobacterium smegmatis to investigate the effect of Aureolic acid group antibiotics. G4 motifs in the genomes of Mycobacterium smegmatis and Mycobacterium tuberculosis were analyzed, of course, and the ability of the Aureolic acid group drugs to stabilize G4 motifs were tested. At the first step, to evaluate the influence of Aureolic acid group on mycobacterial cells, the effect of chromomycin, mitramycin, and olivomycin were analyzed, and BRCA19 was used as control ligands, which describe as G4 ligands. And of course, control cell is cells without drugs. we documented that mitromycin, olivomycin, and chromomycin completely inhibit growth of Mycobacterium smegmatis. In contrast, BRCA19 significantly reduced growth of Mycobacteria only in the first 27 hours. However, by 75 hours, the optical density was close to that the control cells. Those Aureolic acid drugs have antimycobacterial activity against Mycobacterium smegmatis. Moreover, they appear to be more effective than previously described BRCA19. And at the next step, the minimum inhibitory concentration of chromomycin, mitromycin, and olivomycin was determined based on the values of sublethal dose previously reported for these drugs. The minimum inhibitory concentration for olivomycin was 0.5. It should be noted that among antibiotics of Aerobic acid group, olivomycin is the least toxic drug and has the highest hemotherapic index. And the next step, the changes in morphology of olivomycin treated Mycobacterium smegmatis cells were revealed by scanning electron microscopy. On the slide, you can see microphotograph of the cell indicated by letter A, cell control without antibiotics, cultivated without antibiotics, and indicated by the letter B, it's the cell cultivated with antibiotic and different zooms. So as you can see, mycobacterial cells cultivated in the presence of antibiotics were elongated and formed conglomerates not typical for control cells. So really long cell, and these control cells different. And the next step, that is cryptomic analysis, was carried out to determine the mechanism of olivomycin action and drug significant correlation and general influence on the cell, and describe maybe some mechanism of this drug. These data were correlated to this gene's GC content, and we don't find significant correlation between single genes, and while such dependence was documented for promoter operons. And we conclude that olivomycin binds the promoter regions of the operons. And what about specific changes introduced by olivomycin? Mainly concerned defect in cell division. In particular, we observed increased transcription of SOJ gene participating in the formation of cell septum. Previously, it was shown that hyperproduction of this protein leads to disrupt cellular cycle and the formation of treat-like multinucleate cells, which is agreement with our data, which I show on microphotograph. These changes may indicate that the drug is affecting cell division, possibly disrupting the function of that ring and the formation of a septum. Additionally, increased transcription of the FTSY gene involved in cell division and previously positioned as a potential target for antituberculosis drugs. And as conclusion, our data demonstrate antituberculosis activity of olivomycin. We show that it significantly inhibits the growth of Mycobacterium smegmatis. The closest relative of Mycobacterium tuberculosis, transcriptomic analysis revealed a decrease in the transcription of several essential genes. This fact you can see in our manuscript. And we hypothesize that the molecular mechanism of olivomycin activity may be similar to bedaquiline. And all of this allowed us to conclude that olivomycin can be considered as a potential new anti-tuberculosis drugs. I would like to thank my colleagues who worked with me, and of course, thank you for your attention.